How are you and welcome back everyone. I'm today with a new chapter, which is chapter 2. This chapter contains the biological molecules. Somehow this chapter is broader, so I try to uh, classify it into two parts. So in part 1, carbohydrate and lipids are going to be dealt in detail. So please don't forget to encourage me with share, like, and subscribe my channel. Thank you, and let us continue. Chapter 2 of this general biology course is about biological molecules. These biological molecules are referred to as molecules of life, or they are biomolecules that are basically found in living cells. They are categorized as organic and inorganic molecules. The organic biomolecules are protein, carbohydrates, lipids, nucleic acids, as well as vitamins. Whereas inorganic molecules are water, carbon dioxide, and many minerals. These organic molecules contain carbon, hydrogen, and oxygen in some ratio. They must contain these three elements in some ratios, but inorganic molecules have no such elements in some ratios. They may have carbon and hydrogen, they may have carbon and oxygen, and they may have hydrogen and oxygen, but they don't have these three elements at the same time. Carbohydrates, protein, lipids, and nucleic acids are macromolecules. They are required in large amount and they are found in large quantity in the cells, so they are macromolecules. These macromolecules are important structurally as well as functionally for cells. Structurally, because cells are formed or constructed from such macromolecules. Functionally, these macromolecules have a number of functions. For example, carbohydrates can provide cells energy. Proteins can provide cells with repair and growth of the bodies of organisms. Lipids are also energy providers for the cells. Nucleic acids are they are genetic materials of organisms, so they determine the characteristics of organisms. Therefore, these macromolecules are structurally as well as functionally, they are very important for cells. Well, here it is about carbohydrates. Now I'm going to show you some characteristics of carbohydrate and some functions of these macromolecules. So carbohydrate molecules are made up of carbon, hydrogen, and oxygen atoms, with hydrogen to oxygen atoms are found in a ratio of 2 to 1, like that of water. They are with the chemical formula of CH2O in the bracket N. That means N is number of atoms. Carbohydrates are important source of energy. The requirement or the need of organisms are mostly satisfied from carbohydrates. Therefore, they are important source of energy for many organisms. They are most abundant microorganisms, I mean organic molecules in nature. You can think of plants. Most plants are made up of carbohydrates. Therefore, they are most abundant organic molecules in nature than any other organic molecules. And they also provide structural support for cells. as you know, plant cells are, plant cell wall is made up of cellulose, which is carbohydrate. So its function is for supporting purpose. Therefore, these carbohydrates can provide structural support for cells. They help with the communication between cells, or they are important in cell-to-cell communication or cell-to-cell recognition, especially when carbohydrates are combined with protein, which is known as glycoprotein molecules are formed. These glycoprotein molecules are important in cell-to-cell communication or cell-to-cell recognition. Based on the number of sugar units or saccharide they contain, carbohydrates can be categorized into three, or sometimes they are categorized into four. The first category is monosaccharide. These monosaccharides are carbohydrates with one sugar unit or single sugar unit or single saccharide. Disaccharides are another group of carbohydrates which contains or made up of two sugar units or saccharides. Oligosaccharides are carbohydrates that contains few, or especially from 3 to 10 monosaccharides that are covalently linked to each other. Polysaccharides are carbohydrates that are made up of many sugar units. These polysaccharides, oligosaccharides, or disaccharides are made up of from monosaccharides that are linked to each other with a bond known as glycosidic bond or glycosidic linkage. In other ways, carbohydrates are also grouped or categorized as aldehydes, or they are ketones. This category is based on the position of this carbon in carbonyl group. This carbonyl group is a functional group in carbohydrates. Therefore, this carbon in carbonyl group is found as carbon number 1 for aldehydes, or it is found at second position for ketones. Or you can say this carbon in carbonyl group for aldehydes found at carbon number 1, or it is linked with only one chain. These indicates chain. That means this from carbon 2, 3, 4, 5, 6. These carbons and other molecules together make this chain. Whereas in ketones, this carbon in carbonyl group is found between two chains. Can see here, this is oxygen, and here another R or chain is there. We can consider this carbon number 1, and atoms that are found with this carbon number 1 they make you can say R prime, but other carbons from carbon number 3, number 4, number 5, and number 6 together, they make one chain. Therefore, carbon in ketones, especially carbon and carbonyl group for ketones is found in between two chains, whereas carbon in carbonyl group for aldehydes is found or it is linked with only one chain. Or in other words, you can say that in aldehydes, carbon in carbonyl group is considered as carbon number 1, whereas carbon in carbonyl group for ketones is found or it is considered as carbon number 2. This is their difference. As an example, glucose is an aldehyde sugar, whereas fructose is ketose sugar. Okay, and this is about monosaccharides. Monosaccharides are simple sugars because they are made up of monosaccharide or one sugar unit. Based on the number of carbon atoms they contain, based on the number of carbon atoms they contain, these monosaccharides can be grouped as triose, monosaccharides with 3 carbon atoms, tetrose, monosaccharides with 4 carbon atoms, pentose, monosaccharides with 5 carbon atoms, whereas hexones are monosaccharides with 6 carbon atoms. Natherans monosaccharides containing aldehyde groups are classified as aldose, but those containing ketone groups are known as ketose. Aldose are reducing sugar. They are reducing sugars, whereas ketones are non-reducing sugar. Reducing sugars have a capacity to reduce copperic ion, which is copper 2 plus molecule in Bendicase or failing solutions into copperous ion, which is copper with plus charge. As you know, reduction means gaining of electrons. Reduction means gaining of electrons or gaining of hydrogen or losing of oxygen. Whereas oxidation is losing of electrons or gaining of oxygen. Therefore, here copperic ion lose or gain electrons. 1 electron. This copperic ion gains 1 electron. Therefore, its charge is changed from 2 plus to plus 1. Therefore, this copperic ion is reduced. This reaction is done by this sugar, aldose sugars, because aldose monosaccharides have a capacity to reduce copperic ion into copperous ion. In water, pentose monosaccharides with 5 carbon atoms and hexose monosaccharides with 6 carbon atoms exist mainly in the cyclic form. They are found in the cyclic form because this form helps them to occupy a small area. Large amount of saccharides can be stored in a very small area. Therefore, to save their occupation, Therefore, they are found in the cyclic form whenever they are contacted with water. Here I'm going to show you in detail some of the monosaccharides. The first one is glucose. This glucose molecule is aldehyde type. It is hexose and as well as monosaccharide because it is aldehyde. It is this carbon in carbonyl group is found at carbon number 1. It is hexose because it contains, or it is made up of 6 carbon atoms. It is monosaccharide because it is 1 sugar unit. It is reducing sugar because it reduces cupric ion to cuprous ion. So it is what is reducing sugar. This glucose is the most important carbohydrate fuel, or it is most important source of energy, especially for human cells. Therefore, this glucose molecule is taken into the cell and it is metabolized. Then after, energy is released. It is small in its size as well as it is soluble in water. These two natures help glucose to be taken into the cell for energy production, because energy from glucose is produced in the cell. Therefore, glucose must be taken into the cell, passing through cell membrane. For this purpose, these two natures, small size and as well as solubility in water of the glucose molecules, helps glucose to be taken into the cell, passing through cell membrane. The other is two glucose molecules react to each other to make a disaccharide known as maltose, which is malt sugar, sugar found in the malt. These two glucose molecules in maltose are joined together by glycosidic bond. Similarly, starch and cellulose, which are polysaccharides, are made up of glucose. Still, different glucose molecules in these polysaccharides are joined together by glycosidic linkage. Glucose can be found in the cyclic form as well as it can be found in the chain form, especially whenever it is contacted with water. Immediately it will be changed into cyclic form. Here you can see each of these corner or angles are represent the carbon. This is carbon number 1, this is carbon number 2, this is carbon number 3, carbon number 4, carbon number 5, as well as this is carbon number 6. In the chain form, this is carbon number 1 and this one is carbon number 6. Therefore, glucose can be found either in the chain form as well as it can be found in the cyclic form. Here also glucose can be alpha glucose or it can be beta glucose. The main difference of this alpha glucose and the beta glucose is the position of OH group or the position of hydroxyl group of on carbon number 1 of its ring form. Here OH group, which is attached on carbon number 1 for alpha glucose, is found below the plane or below the ring. OH group, which is attached to carbon number 1 of beta glucose, is found above the plane or above the ring. This is the main difference between alpha glucose and beta glucose molecules. Here, galactose is the second monosaccharide. It is aldose sugar, and it is also hexose sugar. It is monosaccharide. It is aldose sugar because the carbon of carbonyl group is found as carbon number 1. It is hexose sugar because it contains 6 carbon atoms. It is already monosaccharide because it's made up of, or it contains 1 sugar unit. It is ready sugar like that of glucose because it has an ability to reduce cupric ion to cuprous ion. This galactose and the glucose have similar molecular structure, but they have main difference. But this difference will be shown on the next slide. Galactose can be found or it can be exist in the form of alpha form or beta form. Galactose can react with glucose to form disaccharide known as lactose. Lactose is disaccharide found in milk. However, galactose and the glucose cannot be converted to one another, even if they have some difference. It is difficult to convert galactose to glucose or glucose to galactose. Okay, this is the main difference between glucose and the galactose. Both have cyclic form or ring form as well as chain form. Still both have alpha form and beta forms. Here beta form, alpha form for both glucose and galactose. But the only difference between glucose and galactose is the position of OH group on carbon number 4. For example, for alpha glucose, the OH group on carbon number 4 is found below the ring. It is found below the ring or below the plane. For alpha galactose, OH group is found above the ring or above the plane. In case of beta form, for beta glucose, the OH group is found still below the ring, whereas for beta galactose, the OH group is found above the ring. For case of chain form, the OH group on carbon number 4 for galactose is found at the left, whereas for this OH group for glucose is found at the right of carbon number 4. This is the main difference between glucose and galactose. Well, fructose is the third hexose monosaccharide. It is ketose sugar because the carbon in carbonyl group here is found between 2 chains. This is one chain and this is another chain. Therefore, the carbon in carbonyl group is found between 2 chains in ketone form. Therefore, fructose is ketose sugar, and it is non-reducing sugar, as that of glucose and galactose. It can't reduce cupric ion to cuprous ion in failing or in NDK solution. It has 5 atoms ring, not 6 atoms ring. As you can see here in the ring form, it makes, or its structure is 5 rings or 5 atom rings. one of this carbon is found out of the ring. Therefore, it makes a pentagon, not hexagon form. This sucrose can react with glucose to form sucrose, which is sugar in table sugar. Ribose and deoxyribose are another monosaccharides. They are monosaccharides because they are made up of, or they contain 5 carbon atoms, so they are pentose sugar. They are monosaccharides. They are pentose sugar because they contain 5 carbon atoms. The main difference between ribose and deoxyribose is the number of oxygen. The number of oxygen. Here for ribose, the number of oxygen is 5, whereas for deoxyribose, the number of oxygen is 4. Their main function is they are parts of nucleic acids or genetic materials. For example, this ribose is part of nucleotides of RNA, whereas deoxyribose is parts of nucleotides of DNA. DNA and RNA are genetic materials or nucleic acids which are made up of nucleotides. Or you can say that nucleotides is monomeric units for this DNA and RNA. However, each nucleotide is made up of 3 different molecules. Each nucleotide for RNA or for DNA, they are made up of 3 different molecules. The first molecule is phosphate, the other is sugar, and the third one is nitrogenous base. But now our focus is on sugar. This sugar is either ribose sugar. For RNA is ribose sugar. It is deoxyribose sugar. For DNA, the nucleotides of DNA contains deoxyribose sugar. However, whenever we discuss the genetic part of this course, we'll discuss this part in detail. Okay, now previously I tried to discuss with you about monosaccharides, especially monosaccharides with 6 carbon atom and monosaccharides with 5 carbon atom. Monosaccharides with 6 carbon atom are glucose, galactose, and fructose. Glucose and galactose are aldose sugar, which are reducing sugar, whereas fructose is ketose, non-reducing sugar. The 5 carbon sugars are ribose and deoxyribose. The only difference between ribose and deoxyribose is the number of oxygen atom. When one oxygen atom is reduced, a deoxyribose molecule is formed. These two molecules, ribose and deoxyribose, are very important for the formation of DNA and RNA, which are nucleic acid or genetic materials. Now, here I'm going to discuss with you again about disaccharides. As the name indicates, disaccharides are, dia means 2, therefore these disaccharides are carbohydrates that are formed from 2 monosaccharides. Actually, monosaccharides are rare in nature. However, most sugars are found in the form of disaccharides. Disaccharides are formed when 2 monosaccharides are react to each other. For instance, when 2 glucose molecules are react to each other, maltose, which is malt sugar, plus water molecule are produced. As you can see here, maltose has 12 carbon, 22 hydrogen, and 11 oxygen atoms, whereas each and individual glucose molecule has 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms individually. But when add up each of these atoms, each of the glucose molecule, it becomes 12 carbon, 24 hydrogen, and 12 oxygen. However, 2 of hydrogen atoms are and 1 oxygen atom are bring together to make hydrogen. I mean water. This water molecule is produced. Such process or such reaction are known as condensation reaction or dehydration reaction, because water is lost, water is liberated in this reaction. To show figuratively, this is the first glucose, and this one is the other or the second glucose molecule. The linkage or the bond between these two glucose molecules is alpha 1,4 linkage or alpha 1,4 glycosidic bond, you can say. This is alpha because these two glucose molecules that react to each other to form maltose are alpha glucose molecules. Because they are alpha, the position of OH group on carbon number 1 for each of these glucose molecules is found below the ring or below the plane. Second, they are 1,4. It indicates the first glucose molecule participates in the linkage. Its carbon number 1 is participated. Carbon number 1 of the first glucose molecule is participated in the linkage. Whereas 4 indicates that the second glucose molecule, its carbon number 4 is participated in the reaction or in the linkage. Therefore, this molecule is said to be, or this bond is said to be alpha 1,4 linkage or alpha 1,4 glycosidic bond. This is about formation of maltose from two glucose molecules. This is another disaccharide, which is lactose, which is milk sugar. It is produced from glucose, one glucose molecule and one galactose molecule by a process known as condensation reaction or dehydration reaction. Here, water molecule is produced or regulated. The other or the third disaccharide is sucrose, which is formed from one glucose molecule and one fructose molecule by a reaction known as condensation reaction or dehydration reaction. Similarly, here water molecule is liberated. Disaccharides are soluble in water, but they are too big to pass through the cell membrane by diffusion. By the way, energy is always produced in the cell. Therefore, the substrate or energy containing molecule must be entered into the cell, passing through cell membrane. However, these disaccharides are, even if they are soluble in water, but their large is relatively big to pass through cell membrane by diffusion. Therefore, it is a must them to break down into their respective monosaccharides in the intestine to pass into blood and then pass into the cell through cell membrane. In this way, disaccharides can be broken down into respective monosaccharides. Then after monosaccharides will be taken into the cell through cell membrane. Then after energy is produced in the cell. So these are, for instance, this is a disaccharide. By adding water, then it will be broken down into two monosaccharides. This is done in the intestine. This process is known as hydrolysis reaction. That means lysing by adding water. It means lysing or breaking down by adding water. Therefore, disaccharides are lysed or broken down into respective monosaccharides by adding water. This process is known as hydrolysis reaction. The reverse reaction is condensation reaction or dehydration reaction, as you can see previously. By hydrolysis reaction, disaccharides are broken down into respective monosaccharides. So in this way, energy can be obtained from disaccharides. Okay, to add some, during hydrolysis reaction, here energy is released, water molecule is consumed, whereas glycosidic bond is formed. Repeat once. During hydrolysis reaction, energy is released, water molecule is consumed, whereas glycosidic bond is formed. In condensation reaction, water molecule is released, energy is required or needed or consumed, and glycosidic bond is formed. Disaccharides are formed by condensation process or condensation reaction, you can say. The three most important disaccharides are sucrose, lactose, and maltose. These are well-known disaccharides. Sucrose is non-reducing sugar as that of fructose, whereas lactose and maltose are reducing sugar like that of glucose and galactose. Here is another type of carbohydrate, polysaccharides. These polysaccharides are formed from many monosaccharides that undergo a series of condensation reaction. When many monosaccharides are undergoes condensation reaction or a series of condensation reaction, polysaccharides are formed. The process is known as condensation polymerization because many glucose molecules or many monosaccharides are participated here in formation of polysaccharides. Building blocks or monomeric units for polysaccharides are called monomers, or can say monosaccharides. That means polysaccharides are built from monosaccharides or monomeric units or monomers. The properties or characteristics of polysaccharides depend on the following factors. For example, it depends on the length. Some polysaccharides may be formed from tens of monomers. Some may be formed from hundreds of monosaccharides. Therefore, the length or the number of these monosaccharides affect or determine the type of polysaccharides. So length is one factor that affects the type of polysaccharides. The other factor is the presence or absence of branch. Some monosaccharides, some polysaccharides are they have no branch, but some polysaccharides may have branches. At some point, they may have branch. Therefore, the presence or absence of branching is also a factor that determines the type of polysaccharide. The other is the presence of foldings. Some polysaccharides may be folded, but some others may be unfolded. Still, there is, if the presence or absence of straight or coiled. These characters is also affect or determine the type of polysaccharides. We'll see in the next slide about these polysaccharides to some extent. Anyways, well-known polysaccharides in nature are starch, glycogen, and cellulose. So in next slide, we'll see to some extent about these polysaccharides. Starch is a polysaccharide which is stored form of carbohydrate in plants. Starch is formed or found where in the plants in the stored form. It is a polymer of alpha glucose molecule, meaning all the monosaccharides that make starch are alpha glucose molecules. It exists in two forms. Either it is it's found in the amylose form, or it's found in the amylopectin form. This amylose is unbranched polymers of alpha glucose molecule, whereas amylopectin is branched polymers of alpha glucose molecule. You can see here amylopectin is branched form, whereas amylose is unbranched form. This is unbranched form, but this one is branched form because it has branches intervally. Glycogen is another polysaccharide. It is amylopectin form, which is branched polymers of alpha glucose molecules. Many alpha glucose molecules are joined together to make glycogen in the branched manner. However, these alpha glucose molecules are formed or came from starch. Starch is the source of these alpha glucose molecules. Then after these alpha glucose molecules are polymerized back to glycogen. Now here, whenever we eat starch-containing food, then after this starch is hydrolyzed into glucose molecules. Then some glucose molecules are passed into the cell and hydrolyzed to generate energy in the form of ATP. However, any excess glucose molecules are polymerized into glycogen. They are polymerized into glycogen molecule. Therefore, whenever we eat starch-containing food, then starch is hydrolyzed into glucose molecules. Then these glucose molecules can be hydrolyzed into energy in the form of ATP, but some other or excess glucose molecules are converted or polymerized into glycogen. Therefore, this glycogen is stored form of carbohydrate in the animal tissue. This is about glycogen. Cellulose, which is a third polysaccharide, which is a polymer of beta-glucose molecules. Hope you remember beta-glucose molecule. In beta-glucose molecule, the OH group on carbon number 1 is found above the ring or above the plane. This cellulose is straight and without any branch. This molecule makes up the cellule of plant cell. Structurally, cellulose is important for the formation of cellule of plant cell. Cellulose cannot be a source of energy for human being because we human beings have no cellulase enzyme which catalyze the hydrolysis reaction of cellulose into glucose molecules. However, herbivorous animals that eat plants can use this cellulose as a source of energy because they have cellulase enzyme. Actually, these animals directly have no this enzyme. Rather, bacteria which are found in the stomach of these animals can produce this cellulase enzyme. Therefore, they mutually benefited from cellulose. The bacteria produce cellulase enzyme, whereas the animal provide the food, which is cellulose or the grass of the plant. Then after the food, the energy produced from this molecule can be commonly used for the plant, the animal, as well as for the bacteria. Well, previously I tried to discuss with you about carbohydrates in detail. Here I'm going to show you some details about lipids. These lipids are esters of fatty acids and alcohol, mainly glycerol. The primary function of lipids is they store energy. Of course, they have various types. These are structure of glycerol and fatty acids. This is glycerol, which is made up of 3 carbon atoms that are linked or that are bonded with OH group and hydrogen atoms. Whereas these are structures of fatty acids. Mainly fatty acids have 2 different parts. The first part is carboxylic acid group, which is a functional group of fatty acids, and the other part is hydrocarbon chain. This is the hydrocarbon chain of fatty acids. Therefore, any fatty acid has 2 main parts: the carboxylic acid group part as well as the hydrocarbon chain part. Fatty acids can be saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are all the carbons in hydrocarbon chains are bonded to each other with single bonds, whereas in saturated fatty acids there are 1 or more double or triple bonds are there. Therefore, if there is 1 or more double or triple bond is there, such fatty acids are known as unsaturated fatty acids. To repeat once, these fatty acids are mostly made up of hydrocarbon chain as well as carboxylic acid group or carboxylic acid group. The fatty acids can be saturated if all the bonds that link carbon atoms in hydrocarbon chains are single, and they are unsaturated fatty acids if at least one double or triple bond is found. Fatty acids with more than one double bond are also known as polyunsaturated fatty acids. This is the structure of fatty acids, so you can see here in the picture. Okay, triglycerides are types of lipids. They can be found in the solid form, or they are fat, or can be found as oil or liquid at room temperature. Meaning triglycerides can be found in the fat form, or they can be found in the oil form. As you know, fat is solid at room temperature and oil is liquid at room temperature. These triglycerides are stored in adipose or lipocyte cells. This is to facilitate energy store in animals' body. I'll try to show in the next slide what adiposits or lipocyte cells are. In triglyceride, this glycerol and the 3 fatty acids are joined together by a bond known as ester bond. One glycerol with 3 fatty acids, they make a triglyceride. The bond is known as ester bond. This ester bond is formed in this way. This OH group on glycerol are totally removed from carbon, or they are detached from each carbon. Immediately the bond that join carbon with OH group is open. Thus the hydrogen atom of the OH group of the carboxylic acid group of fatty acids are also detached from oxygen. So immediately these carbons on glycerol are joined together with oxygen, which is part of OH group of carboxylic acid group of fatty acid. Already each oxygen is attached with carbon, and after the lost OH group from glycerol and the lost hydrogen from OH group of carboxylic acid group immediately join together and make water molecules. Water molecules are formed, 3 water molecules, because from this 3 OH group of glycerol and 1 hydrogen atom from each of the OH group, immediately they are joined together to make 3 water molecules. So to sum up, from 1 glycerol and the 3 fatty acids, immediately 1 triglyceride molecule is formed and 3 water molecules are liberated. The process is known as condensation reaction. If 3 water molecules are added on triglyceride molecule, immediately 1 glycerol and the 3 fatty acids are produced back. Adipocytes or lipocyte cells are known as fat cells. These fat cells are categorized as white fat cells or brown fat cells. The difference between them is a way of storing lipids. For example, these white fat cells store one large lipid droplet. For example, if this is white fat cell, it stores 1 droplet of fat, large droplet of fat, whereas the brown fat cells stored smaller and multiple droplets of fat throughout the cell, like this. If this is a brown fat cells, it stores smaller but multiple fat droplets. This is their difference. White fat cells stored one large lipid droplet, whereas the brown fat cells stored smaller but multiple droplets of fats throughout the cell. Okay, these are polar lipids. For example, phospholipid is one of the polar lipids. It is formed from one glycerol. This is like if you take as a glycerol, and this phospholipid is formed from one glycerol, two fatty acids, and one phosphate group. If this is a fatty acid, one, and this one is a second fatty acid, then the third carbon is attached with phosphate group. So one glycerol, two fatty acids, and one phosphate group can make phospholipid. Mainly this phospholipid is very important, or it is main part of cell membrane. By the way, we'll see in detail about this molecule whenever we are discussing about cell membrane. Therefore, phospholipid is major components of the cell membrane. The other lipid type is glycolipid. This glycolipid is lipid type when carbohydrates are attached or joined with lipids and mainly associated with the cell too, as that of phospholipid. But its main function is it provides energy, and it serves as marker for cellular recognition, because in level of organization, similar cells come together to make tissue. Therefore, for that purpose, cells must be recognized to each other. Therefore, this glycolipid, which is part of the cell, helps these cells to be recognized to each other and to make the respective tissue. The other lipid or polar lipid type is sphingolipid. This sphingolipid is a lipid type which is formed between fatty acids and sphingosine. Instead of glycerol, there is sphingosine is attached with different fatty acids. The other lipid type is wax. Wax is found especially on the leaves of plants, especially at the desert area. Plant leaves are covered with this wax, which helps them to prevent water loss. Even honeycombs in bean is also made up of bean wax. All these are types of polar lipids. Well, cholesterol is a type of blood fat, and blood fats are already known as lipids. Therefore, cholesterol is a type of lipids. This cholesterol is a type of steroid types, which contains 4 fused carbocyclic ring system. This is structure of cholesterol. This structure or cholesterol structure has some special features. For example, this cholesterol is 4 fused carbocyclic ring system. This one is the first ring, the second ring, third ring, and the fourth ring. This cholesterol is formed from 27 carbon atoms that are joined together and with other molecules. Plus this cholesterol in third carbon OH group is attached. This is a special feature of cholesterol because this OH group will help the cholesterol to esterified with different molecules. Plus, this cholesterol has double bond in 5 and the 6 carbons between 5 and the 5th and the 6th carbons. Whenever we think of cholesterol, immediately in our mind we think that cholesterol is a harmful molecule. Of course, if the requirement is more, or the concentration of cholesterol in our blood is more, it may cause for different effect in our body. It may block blood vessels, and it may block the flow of blood through the blood vessels. However, in appropriate amount, cholesterol has a number of functions in our body. For example, it is a component or part of cell membrane. The fluidity of cell membrane is determined by the by cholesterol. When the amount of cholesterol increases in the cell membrane, the fluidity of cell membrane decreases. But when the amount of cholesterol decreases in the cell membrane, the fluidity of cell membrane increases. So the fluidity of cell membrane is determined by the concentration of cholesterol. Second, cholesterol can be used in nerve condition because cholesterol insulates the nerve fibers. It can be used to make bile salts and bile acids. These bile salts are important for fat absorption. Whenever we eat fatty food, immediately they are broken down into smaller droplets by bile salts. Then after, enzymes are act upon them. And also, this cholesterol is important molecule for the formation of steroid hormones, especially glucocorticoids, androids, and estrogens are hormones, especially steroid hormones. And for the formation of these hormones, cholesterol is important. Especially androgen and progesterone are hormones that control secondary characteristics of male and female respectively. Vitamin D3 is formed from 7-dehydrocholesterol 2. Esterification is also occurred. The process of esterification can be occurred with cholesterol because cholesterol has OH group on its third carbon. Therefore, whenever it reacts with different molecules, maybe ester bond is formed. Therefore, this esterification is formed between cholesterol and different molecules. Properties of lipids. Lipids are insoluble in water because they are, they are nonpolar molecules. They are longer in chain because fatty acids are mostly longer, so the whole molecule is longer in chain. Double bonds increase solubility. Lipids that contain double bond, fatty acids that contain double bond are unsaturated fatty acids. So these lipids that contain unsaturated fatty acids are more soluble than that of unsaturated fatty acids. Melting point depends on the length and saturation. Saturation. That means the presence or absence of double bonds in their fatty acid part. In unsaturated fatty acids have lower melting point, the saturated fatty acids. These unsaturated fatty acids are liquid at room temperature, more of their oil, and saturated fatty acids are solid at room temperature, mostly they are fats. But it is possible to convert these unsaturated lipids or fats into saturated one by a process known as hydrogenation. By adding hydrogen, it is possible to break the double bonds and make single bonds. These are some importance of lipids. They are the main components of cell membrane. Cell membrane is mainly made up of lipid known as the phospholipid, and it also has some amount of cholesterol, which determines its fluidity. Then lipids are important in insulation of heat and water. In cold environmental condition, organisms may lose their heat. So to prevent the loss of heat, these fats insulate heat, and in warm environmental condition, organisms may lose their water. So not to lose their water, these lipids prevent the loss of water. So they insulate the heat and water. And lipids store a large amount of energy. Even they contain twice as much as that of carbohydrates. So they contain or they store energy. They used as a protection as well as cellular communication. For example, the nerve cell is protected by lipids, and even they are important in cell-to-cell communication. For example, when they are joined with protein, lipoprotein molecules are formed. So these lipoprotein molecules are used for cell-to-cell communication. So these are some functions of lipids.